Today's topic is the concept of recombinant deoxyribonucleic acid. Methods of obtaining recombinant deoxyribonucleic acid. The purpose of training. Explanation of methods of obtaining recombinant deoxyribonucleic acid. The main meaning of genetic engineering is the task of creating a recombinant DNA system. Recombinant DNA is understood as DNA formed by combining any two or more DNA fragments that are different from the source of origin in vitro. The simplest composition of recombinant DNA consists of free engine DNA and a vector. The most common methods of compiling recombinant DNA relies on the use of restrict synthesis. This is called the sticky chip method. In this method, recombinant DNA assembly is performed using the Cohen-Bayer model. Again, also a fragment of free DNA and a vector are interrupted by a single restricted enzyme, as a result of which their ends easily merge with each other with the help of the DNA ligase enzyme into gibbered or generic DNA molecules since they are complementary, that is sticky. The adhesive tip method relies on the restrict traces that break a piece of the DNA that is recognized at some distance from the symmetry grows. Recommended DNA. Recommended DNA assembly by the adhesive and the method has its pros and cons, and the resulting graded DNA sequence as a restoration of parts that can be recognized by retase characterizes the good side of the method. This makes it possible to isolate a fragment of region the and relatable easily under the influence of these restrictions. Well, the fact is that all the chains obtained by recession at adhesive and are rejoined with each other homologous chains indicates a disadvantage of the method. As a result, a number of vector molecules are recruited not by embedding but by direct interaction with their own ends, while other vectors can merge with embedding made up of several combined foreign DNA molecules. Therefore, it is necessary to carry out work on the selection of hybrid vectors with only one embedding. Recombinant DNA. The main content of genetic engineering is to separate the necessary gene in the body and combine it with a vector. Here most often as a genetic vector is plasmid DNA. The resulting hybrid DNA molecules is embedded in the bacterial cell. As a result of the doubling replication of vectors in a bacterial cell, the number of prepared gibbered DNA molecules also begins to increase. And this process is called recombinant DNA cloning. Vectors are the genetic structure necessary to transport genes from a test tube to a biological laboratory and ensure their normal functioning. We know that plasmids and phages can act as vectors. The fact that plasmids are resistant to any antibiotics makes it possible for bacteria to be selected for such antibiotics, and because the recombinant DNA molecule is easily identified, they are considered good vectors. Before entering a bacterial cell, it is necessary that the DNA segment gain intended for cloning at the molecular level is capable of replication. However, it does not have this ability, and therefore it combines them with genetic vectors to ensure that they are transported to the cell and that the clone genes in the cell remain known. Along with plasmids, virus, bacteriophages, animal and plant viruses can also be used as vectors. However, due to the fact that animal and plant cells have some differences in the composition of chromosomes and the behavior of cancer bacteria, it is more difficult to perform genetic engineering manipulations on them.